So what's the fastest way to determine if an agonist's potency comes from affinity versus efficacy, or both? Well, it's usually both, but there are compounds that have preferential. So that if your agonist depends on affinity to be potent, usually what happens is its efficacy may be in not enough to give you response in a poorly sensitive tissue. And so what happens is a affinity dominant agonist will be very tissue selective. That is, it'll give you responses in a sensitive tissue, but in one that is not as sensitive, you will get much less response. Whereas an efficacy dominant will produce responses in all systems. Now, natural physiology, transmitters and so forth, nature pretty well made them all efficacy dependent, probably because you need responses in all tissues. And so things like epinephrine, acetylcholine, and so forth have very poor affinities, but very high efficacies, but they will produce responses in all systems. There are cases where you might want an affinity dominant compound. So I'm going to tell you a quick story about alpha receptor constrictors. So these constrict arteries and veins, and you can have affinity dominant ones that will preferentially constrict capillaries and small arteries and veins over large vessels such as aorta and so forth. Why might you want that? Well, if you have shock. So now in shock, all your arteries and veins are relaxed, blood pressure is low. The way to get it up, of course, is to give a pressor agent, something that will constrict the alpha receptors in an artery and vein, constrict vessels, elevate pressure. But if you do that to all vessels equally, what you tend to get is steel. So you're going to also constrict vessels to the organs you want to get preferential blood flow during shock. Whereas an affinity dominant will constrict the most sensitive vessels, which are the smaller ones, pumping blood into the larger ones, and that's what you want. So it gives you a preferential constriction where you would need it. So it can be important. In most cases, when you're dealing in a program and you have an agonist, programs often use a very sensitive receptor overexpressed system, and they get nice agonist curves. Without knowing if that's due to a high efficacy or a high affinity, you might get a nasty surprise in vivo or in a different system that is not as sensitive, because with a highly sensitive system, you won't know if that potency is primarily due to affinity or efficacy. So you take those and you test them in a less sensitive system to check that.
Thank you for that, Terry. And if I can quickly jump in, I think when you're using that very high expressing system, that very sensitive system, especially in the beginning of a program, you are just happy to be able to detect something from the molecules that you're testing. And in fact, there is a question that came in, and the question is related to system and receptor expression. And the question goes as follows: when should a team intentionally use high and low receptor expression to expose hidden drug behavior?
Great question. I call this tissue volume control. Consider your system as a radio, and it has a control. You can have it loud or soft. Very useful to have both, have a high expression system and a low expression system. The high expression, good for screening, sensitive and so forth, good to demonstrate agonism, but also good to pick up low efficacies in an antagonist. Because if you don't have a sensitive system and test the antagonist only in a normal or low sensitivity system, you may not detect an efficacy that might be unveiled in vivo. So you want to check that. Go to a high expression system and see if your so-called silent antagonist isn't silent, but actually produces response. Low sensitivity systems are extremely useful. We just talked about detecting whether agonism potency is due to high efficacy or high affinity. That's one way to do it. That'll be revealed in a low sensitivity system. But more than that, that's a great system to work out relative efficacies and affinities of agonists. You can apply the operational model there and get actual numbers that you cannot get in a highly sensitive system. It's also easier to measure affinities in a low expression system. So, if possible, the ideal world is to have a cell system where you can induce levels of receptor, low and high. Now you've got it all. You've got a system that's useful for all things, and it's just one cell line, which is nice.